In the previous part, we finished by discussing the mechanics and the mechanism of the skeletal muscle contraction, and so we looked at those three kind of general events and phases. So what happens at the neuromuscular junction, what happens at the sarcolemma, T-tubule, sarcoplasmic reticulum, and then finally what happens at the level of the sarcomere itself, right? So contraction can actually continue or the process of contraction can actually continue into death. And so this is referred to as rigor mortis. So the term rigor in this case is referring to stiffness or firmness, and then mortis is, of course, referring to the idea of mortality or death. So rigor mortis means the stiffness of death. And so this is what is occurring in animals, humans included, post mortem. And we have this kind of quick, rapid contraction that can be somewhat sustained and it onsets at some point after death. It can onset a number of hours after death, and it usually disappears somewhere maybe between 24 and 48 hours after death. And so what you have happening is you essentially have the same mechanics of contraction occurring post-mortem that you had occurring ante-mortem, so before death. So what you have is as you die, or not as you die, but when you are dead, you have the breakdown of cells, and inside cells, you have the breakdown of the organelles within cells. One of the organelles, in particular for our skeletal muscle, is the sarcoplasmic reticulum. And so whenever we break down the sarcoplasmic reticulum, you release the calcium out of the SR and it enters into the sarcoplasm. Now, this calcium can wander around and it can ultimately find its way to troponin, as we mentioned in previous parts of this lecture. And when it binds troponin, calcium is serving here as the lock and troponin is the key. And when you bind calcium to troponin, you expose the active sites or the binding sites for the myosin head. And so if you then have ATP, you're able to have the myosin head bind, and then you can actually get a contraction. So you can get a contraction after death, right? So when you're dead, you still make a little bit of energy. You still make a little bit of ATP via aerobic respiration. Via anaerobic respiration without oxygen present. And so you can have oxygen present, you do not have oxygen present. You still make a little bit of ATP. And this ATP then binds to the myosin head. It's hydrolyzed, it's split. This provides energy for the cross bridge formation, and then the phosphate leaves and you get the power stroke, right? So this can continue happening and you can continue to have contraction or relaxation. After death, at some point, you may have calcium still there, and you may have enough ATP to have a contraction take place, but then you no longer have ATP to cause the relaxation, right? So if you recall, we mentioned how ATP is used for both contraction and relaxation of the muscle. And so if you have the contraction, because you had ATP and calcium, but then you use up and you expend all of your ATP, you now have no way to relax your muscle. And this is what you recognize as rigor mortis, right? So if you drive along the highway, or you drive along the interstate, oftentimes you'll see dead animals, you'll see a dead deer, or a dead pig or something. And if it's a more fresh specimen, if it was hit more recently, then you may see its legs, you may see its extremities kind of sticking out to the side in a very rigid manner. This animal has undergone rigor mortis, right? So it's kind of twofold. It's due to the calcium, and it's also due to the lack of ATP at some point after death, all right? So if we look at our contraction cycle, we can see that there's a number of different phases of our cycle, all right? And these are corresponding to what is happening to the muscle and then what is happening to the amount of tension that the muscle is generating. All right. So you have three general phases. You have what's called the latent phase, the contraction phase or period, and then the relaxation period. So in the latent phase, the term latent is referring to kind of resting or quiet. And so in this case, we're starting our muscle contraction, but we have yet to generate any tension, right? We have no tension generated. So this is the time whenever the action potential is traveling along the sarcolemma. It goes along the sarcolemma with sodium going out, sodium coming in, potassium going out. It then travels down the T-tubule or the transverse tubule, and it passes by the voltage-gated calcium channels of the sarcoplasmic reticulum and stimulates them to open to release calcium. Calcium has been released, but in this phase, in this latent phase, calcium does not yet bind to troponin, right? So if you don't have calcium bound to troponin, you can't have the binding sites for the myosin head exposed, So you can't have cross-bridge formation. If you don't have cross-bridge formation, you can't have the power stroke. And if you don't have the power stroke, you can't have contraction and tension. So if you look at this first phase here in the latent phase, you see we're not generating any tension. We're staying relatively flat, all right? So if we move to the next phase, we move to the actual contraction phase, right? So the contraction phase is when we actually begin active contraction. Calcium has now it is now bound to troponin. When you bind calcium to troponin, you shift that troponin tropomyosin complex. You expose the binding site for the myosin head. You get a cross-bridge formation, and then if ATP is present, you get your power stroke, you get your actual contraction, right? So as you generate contraction, you generate tension, right? So here we begin increasing and increasing in tension to some peak point, and then we enter into our relaxation period, relaxation phase, where we begin tapering off this tension, right? So we begin dropping. This occurs as acetylcholine is being broken down at the neuromuscular junction. Cross bridges are being broken by a new ATP coming and binding to the myosin head. You have calcium being pumped back into the sarcoplasmic reticulum. So this calcium, if we focus on this for just a second, the calcium, when it left the sarcoplasmic reticulum earlier, it leaves via passive transport, right? It just goes from high concentration to low concentration out of the sarcoplasmic reticulum into the sarcoplasm. So no energy is required. However, during relaxation, whenever we are trying to pump the calcium or we're trying to get the calcium back into the sarcoplasmic reticulum, you have to do so actively. So meaning you have to use ATP because this is a form of active transport. So if this is active transport, this tells us that we're going against a concentration gradient, and that's exactly what's happening. So although calcium rushed from the sarcoplasmic reticulum out into the sarcoplasm, there is still more calcium in the sarcoplasmic reticulum than there is in the sarcoplasm. So by putting the calcium back into the SR, you're having to go against its concentration gradient, so you have to use ATP, you have to use active transport to do so, right? So we can take this muscle twitch. So one muscle twitch is the latent period leading up to contraction, the actual contraction, you're generating tension, and then the period of relaxation. That is one twitch, right? So we can put these twitches together, and when you do that, you're essentially summating the twitches, all right? So just like in the previous lecture in Chapter 8, we talked about the summation of graded potentials, so EPSPs and IPSPs. You can do something very similar here, all right? So you have two types, and these are referred to as tetany. This is kind of from the same term as tetanus. If you have a tetanus shot, right, it can cause contraction of a muscle. So in this case, we're looking at the contraction of a muscle, and in particular, we're looking at the amount of relaxation after one muscle twitch, all right? And so these two types differ in the amount of relaxation. So you have what's called incomplete tetany or unfused, and you have complete tetany or fused tetany, right? So if we start with incomplete, you notice that it resembles a kind of staircase model. So you have some peak amount of tension. So these rising phases here correspond to these rising phases that you see here, all right? The decreasing phases that you see here, where tension is dropping, correspond to the period of relaxation here on this muscle twitch diagram, right? So in this incomplete tetany, or in incomplete tetanus, you notice that you generate tension, you increase, and then you begin relaxing. So you begin decreasing tension, but you don't decrease the tension all the way to where you started, right? So we started here with, say, zero tension. We came up to some peak tension here, and we begin relaxing, but we didn't relax all the way. So what this means is that we received another stimulus. There was another action potential that arrived. We got another depolarization, repolarization, cross-bridge formation, power stroke, all of that. And so the muscle contracts again. So you get more tension, right? So you have more tension, and then you begin relaxing. And then you have another one, and you have more tension, and you relax again, so on and so forth, all the way up to some peak amount of tension, all right? So the reason we don't have just an exponential increase in tension, and it doesn't just go on continuously, the reason there's a maximum is because we only have so many myosin heads to form so many cross bridges, to perform so many power strokes, and we only have so much calcium to bind to so many troponins to expose so many myosin head binding sites. So this means that we are limited in the amount of tension that we can generate, right? So theoretically, if there were an infinite number of myosin heads and an infinite number of myosin head binding sites and an infinite number of infinite amount of calcium, then I suppose theoretically you could have some infinite amount of tension that you can generate, but that's not the case, right? So you note that you begin increasing tension from here to here, but then this jump from this tension to this peak tension is greater than this tension to this one. And likewise, the tension from this point to this point is greater than the tension from this point to this point. And so what you have is you have this kind of increasing amount of tension, but then it starts going the opposite way. So the tension between this point and this point is less than the tension between this one, and likewise the tension between this one and this peak amount of tension is less than the tension between this point and this point here. And so as you go up, you are, again, you're forming more cross bridges, and so the more cross bridges you form, the less you have to form in the future because, again, we only have so many. And so you see this beginning to kind of increase at the beginning, but then as toward the end, it starts to taper off until you get some peak amount of tension, right? So if you understand incomplete tetanus, complete tetanus is exactly the same thing, except you don't have any period of relaxation, right? So you start here with no tension, right? Just like we started here, and you begin increasing, but you note that there's no period of relaxation. There's no drop off in the tension. You just continue all the way up, but again, only to some maximum amount of tension, okay? So we can also look at our muscles in terms of the muscle tone, right? So muscle tone is referring to the amount of tension that we see kind of at rest. And so this results from the number of what are called motor units. And so a motor unit is one neuron, one motor neuron, innervating all of its respective muscle fibers, right? So in one muscle, there's lots of muscle fibers, and say 10, 15, 20, however many different muscle fibers are innervated by one motor unit, all right? And so you have different muscles that have different motor units. And so in muscle tone, you have a motor unit that is being innervated, and it can have some degree of kind of constant involuntary innervation, right? So this results in a resting tone or kind of some resting tension in the muscle, right? So if you look at Arnold here, if you were to touch his biceps or abs or pectoralis major muscle or deltoid or traps or something, at rest when he's not contracting, it would probably feel relatively firm. It would feel as though it was contracted because you have this small amount, or in this case, a larger amount of resting tension, all right? You also have the size of the muscle itself. So whenever we lift weights, whenever we do some type of resistance training and we grow our muscles, the muscles are getting larger, but the individual muscle fibers are not. All right. I'm sorry. They are getting bigger. They're not increasing in number. Okay. Excuse me. So I misspoke. So I want to make sure you understand this. The muscle cells are not multiplying, so we're not getting more cells, but the cells themselves are getting larger, all right? We pack in more and more myofilaments, more myofibrils. This increases the size of the cell, so the muscle itself appears larger, but we're not increasing the number of cells, right? So this is referred to as muscular hypertrophy. So hypertrophy means a cell gets larger, so in this case, the muscles are getting larger. So there's two types of hypertrophy. Generally, you can think of physiological hypertrophy or non-physiological hypertrophy. So physiological hypertrophy means it's normal, part of some normal physiological process, like lifting weights, some type of resistance training. Non-physiological muscle hypertrophy would be something like taking anabolic steroids, right? So you still get the hypertrophy, you still get the increase in muscle mass and perhaps muscle strength, but you're doing it artificially. It's not normal physiological muscle hypertrophy, right? And then the third factor that can influence muscle tone is it's not really influencing muscle tone, but it can appear as though the tone has changed. And this has to do with the amount of subcutaneous tissue around a muscle. So essentially, how much fat is around a muscle can influence how that muscle appears from a very superficial view, right? And so the less fat you have around a muscle, the more that muscle is going to be shown. And so it may appear as though it's very toned, whereas in reality, it's just you see it more because you now no longer have that additional adipose tissue around it, okay? Another important aspect of muscle is the length of the muscle, all right? So there's an important relationship between the length of a muscle as well as the amount of tension that you can generate, all right? And it is not the case that as you continue to increase the muscle, you continue to increase the amount of tension. You do up to a point, but not continually, all right? So here we have a muscle that is 70% of its optimal length. Here we have, and this is for skeletal muscles specifically, here we have muscle at resting length, so 100%, and here you have muscles that are stretched beyond kind of a normal resting length, right? So the length of the muscle influences the amount of overlap between actin and myosin, right? So when actin and myosin are overlapped, you get some number of cross bridges that can be formed, right? When you get so many cross bridges that are formed, you get a certain number of power strokes, and these power strokes equate to so much tension, right? So for skeletal muscle, at rest, our muscles are typically at the optimal length to generate the amount of tension necessary, or the maximum amount of tension necessary, right? Now, we don't always generate that maximum amount of tension, but we have the ability to, okay? So here we're at rest, 100% perfect, and we have this amount of overlap, right? So these green filaments are the actin, and these red ones here are the myosin, okay? You can see here, if we go to the left now, we're shorter. You have more overlap, but some of the actin is not actually going to be able to form a connection with the myosin. And so this means that if you have less myosin heads that combine actin, you get less tension, or less cross bridges, less power strokes, so ultimately you get less tension, right? If you move the opposite way, you begin stretching the muscle even longer, so this sarcomere has elongated. You've now lost some of the overlap here, and say now you can only form, in this example, maybe you only form 2 cross bridges or 3, as opposed to forming maybe 4 or 5 here, right? And so fewer cross bridges, again, fewer power strokes, less tension. And then same thing if you increase all the way to the point where you have no overlap between actin and myosin, you generate no tension, right? So this is also important in the context of cardiac muscle tissue, but cardiac muscle tissue is a little bit different. It is not at its optimal length to generate as much tension because the amount of blood in the cardiovascular system and the amount of blood returning to the heart can change. And so the heart needs to be able to adapt to that change in blood volume to generate more tension so it can generate, or it can then pump out that additional amount of blood. All right. So in the context of the heart, this is referred to as Frank Starling's law of the heart, or just Starling's law. And it reflects how an additional amount of blood called preload, the stretch of the ventricle in the heart, can actually lead to an increased force of the contraction, so an increase in tension. All right, so we will stop here with this part, and we'll pick up with the next part, talking about how we get our energy for all of this muscle contraction that we've been talking about.